So let's get into it. See, how can I get this out of the... Okay, I am Matthew Koons, and I'm an amateur mycologist. I'm kind of focused on field work. And while I am a huge appreciator of broader biodiversity, arthropods, plants, anything that I can take a nice picture of while I'm out on the trail, I'm really focused on surveying for mycodiversity and collecting and vouchering, and now since last September, doing some barcoding out of my own house, which has been a journey. I know you have to have a photo with a mushroom, so I put this fully picture. I'm going to cover some assumptions for this talk before I move too far forward. I'm going to be wrong at least once, if not a bunch of times. I think there's so much to make a true statement about some of this. And I will also be hypocritical because I'm going to teach you about some best practices for iNaturalist and ask you to make a lot of observation fields that I don't do that on every single observation by a long shot. So really just going to cover some basic concepts to kind of make a foundational understanding, and then we'll move forward with some of the more detailed things about collection. And also please resist the idea that you would need to buy anything to just get out and naturalize and look at mushrooms. I'm going to show you things that I use. It doesn't mean that you need to have them. You can just go out with a paper bag and learn as much as I do with camera equipment and tackle and all of this.
So we see who's the star of the show.
Okay, well, it's time for me to come clean how I first became interested in mycology was not a cute mushroom that I saw on the side of the road. I was in college. I encountered a psilocybe in a little bag, and it really opened up my perspective on what was possible as far as what my mind was capable of, what, you know, if you've interacted with this group of fungi, I think you know pretty much what I'm talking about. There's a lot to learn. There's a lot to be shown from the fungal kingdom, and these speak the most directly to our psychology, I think, in a lot of ways. Anyone can have an impact on our regional knowledge of mycodiversity with just a camera, a local park, and iNaturalist. All these are things that I found just in parks around Seattle, which just goes to show you don't have to go deep into the mountains. You don't have to go out of the country to find something interesting. How can I move this bar out of the way?
Just try to include.
I don't know what to do with this. It's blocking some of the stuff on the screen. We'll just have to deal with it. Let's see. Is that the Zoom by?
Yeah, there should be
hide. Which one should I pick? Floating meeting controls? Yes. We've done it. Okay, this is my first time presenting any sort of mycological talk. So this is really my first huge hurdle of getting into my talk. This pseudobayospora took me 3 years to get to the bottom of what it was. My friend Connor Dooley put me on the right track after I put some woefully wrong iNaturalist species up there. Onogena corvina really only grows on decaying birds. It's hard to find. If you come across it, it's really a special fungi. I think you're really supposed to limit how much you handle it because it attacks, what's the substance in hair in your fingernails? Keratin. It's keratinaceous. So like, it could get into your cuticles or something like that. But in this same family is athlete's foot. So we don't really know exactly what's going on there. This Lepiota is how I first got in touch with like the first thing I sent in for sequencing ever is through PSMS. I put it up as an echinoderma. People were like, we don't have that here. And then Danny Miller reached out to me. He's like, can you send me this? That was the first time I ever talked to Danny, who is a regional barcoding, icon essentially. And then we send it off. It comes back. It's actually a Lepiota species that looks just like Echinodermas in a lot of ways, with these kind of warty, ornaments all over. So let's keep moving forward. How do I click? You want to find interesting fungi, and so do I. So where should we go? If you don't have a whole day to drive out to the mountains, just go to the nearest park from you. I live about right here. All of these parks are within 15-minute drive to an hour's walk, and I've found undescribed species here, here, and here, not so far in this park. But if you also want to find a certain thing, you want to find Boletus edulis or whatever, you can plug that in to iNaturalist and all of the locations will show up. You can just go there during the time of year that it happens, and maybe you'll find it. I used Mitchell as an example because I did this with Noah Siegel's observations in Mitchell. I wanted to see them. Couldn't find them on my own after I tried for a year or two, so I just kind of cheated. I want to talk briefly about mapping because you're going to want to go outside of town eventually. These green trails maps are really great if you're going to be in the backcountry for a few days. You don't want to trust batteries on your phone because they're just solar powered. They're just paper maps. You don't have to worry about battery. But I highly suggest that you use a GPS app if you're going to go into the backcountry, especially alone. There's a lot of options down here that I've tried to include. The only things that really matter for me are that you can save a map offline. You can download the area that you're going as well as your active track, so you can come back the same way that you went if you want to. And then reliable elevation. A lot of times I just leave my location data turned off on my phone in the backcountry to save battery. And I'll just take screen captures of my GPS mapping and use that for elevations and all things when I'm making observations later. And just to touch on safety briefly, because I don't really have another opportunity, like go with someone else if you can. Buy a water filter and just carry it with you all the time. That's the main thing. Carry enough food to get you through the night, even if you're not planning on going overnight. Look at the 10 necessities for backcountry survival and do your own thing there. Similarly, permitting is really complicated, and I'm not going to try to break it all down for you, but something you should be aware of, like whether it's legal for you to collect fungi where you're going. The best way to do that, as it says there, is to just go by a ranger station. They're usually really kind. They're going to be really interested in what you're doing. They'll be very helpful, let you know exactly what you can collect. A lot of times it's a 5-gallon bucket or something like that per day. And for what I'm doing scientifically, just collecting small mushrooms here and there, I've never come close to that. And you will need these 2 passes at most trailheads in the Northwest. The Discover Pass is for Washington state lands, like if you go out to Deception Pass or something near here, you'll need a Discovery Pass. Forest passes cover national forest, which is mostly montane, but there's localities around here where you need a forest pass. And the America the Beautiful Pass that you can use to go to national parks is a Forest Service pass that's higher than the National Forest Pass, so you can use that for dispersed camping. Does anybody do any dispersed camping here? Okay, it's really the best way to go camping by a long shot. There's no bathrooms, but that's really the only con. And if you're fine with digging a hole there, there's no cons. It's a lot of campsites stretched out along Forest Service roads. You'll see little pullouts with old fire pits, and you can pull into any one of those. You'll be right next to the trailhead that you want to go to. You'll get to see all kinds of plants, all kinds of animals, birds, arthropods, whatever, just walking through your campsite. And then in the morning, you'll be able to get to your campsite or get to your trailhead way before anyone from the city can come out there. So I have a little bit more to read on this one. So fungi can't move, but you can. They're effectively just waiting for you to be in the same place as they are and observe them. The best way to do that is when you're in one of these environments, just to pause and crouch down every once in a while and just sit there for a few moments and sort of start to absorb what's coming out visually from the background of the trail dust that you've been walking past. And a lot of things will start to poke out to you, little ascomycetes, all the tiny things that I've been photographing that are in my opening slides. Most of those I've just found by stopping to look at something else while I'm down there. I see something smaller. While I'm looking at that, I see something even smaller. And then we've been there for an hour and a half, and my girlfriend and my dog want to keep walking. We got Jack Johnson and Lauren Ray here in this photo. And I think they're the first people in this presentation to get a gold star, which is going to be a theme. Essentially everyone I've met and mycologized with is going to get a gold star tonight. So here we go. Let's see what else we got. So we're going to use all of our fungus, or all of our senses essentially when we're trying to observe fungi in the landscape. Hearing is the least involved, but there are some ascomycetes that hiss a little bit whenever they eject spores. And if you're walking up to a wetland environment, you might hear some trickling water. You be tuned in, you're more likely to find something in the summertime. And making a habit of spending time outdoors will help you tune in to seasonal cycles and phenology, which is a fancy way of just saying seasonality with plants, fungi, all of the ecological systems that work together and make being outdoors so rewarding. But we will, like I said, we're going to use all five senses. So we'll use touch a lot, tell you how firm a mushroom fruit body is, or all kinds of other textural information. We can use taste. You can taste safely mushrooms, even that are toxic, as long as you spit them out afterwards. A lot of things you'll want to have water handy to rinse your mouth out with. A lot of acrid russulas and all kinds of other things that actually taste really spicy or generally pretty terrible. So, and smell is also part of that. Taste and smell are kind of tied together. But another metaphor I use for hearing is for barcoding, because we're picking up on latent frequencies and we're translating them into a visual form. It's kind of like hearing. I don't know. So what I meant to say before I transitioned over to this slide is, in all of those different microclimates that were pictured, a lot of them have really limited ranges of fungi or plant communities that can exist there because of biotic and abiotic factors. One of my favorite is peatland, which is going to be a high acidity environment. They can happen down here at sea level, or they can be in montane environments where they're usually called fens. And these are just a lot of fungi that you can find here. If there's any photos that are not mine in this talk, they're going to have a little white attribution here. This photo is by Connor Dooley. You can find a lot of these fungi actually pretty close to Bellingham. Hygroscope tarunda is going to be in just about every wetland you come across. Hygroscope substrangulata is even more common, even though I've only seen it a few times. If you go south from here, it seems like it's unavoidable. Umbrophylla, Connor and Jack and I found together in a fen out the north side of the North Cascades last summer, which was a treat. Really, a lot of these you're going to see in a lot of different environments. Let me move my name out of here because this one is Gymnopolis fulgens, which you can find outside of a wetland fen environment. And this is kind of a morphological outlier for this species, but genetically they're identical. So they're really just growing on compressed sphagnum, but you can see here in the late summer is when I've seen them after a lot of the water is drained out of these wetland environments. We have fungiculus fungi, which is fungi growing on fungi. A lot of you must know Jack Johnson. He has like organisms on organisms Instagram, and this kind of reminds me of that. And really, I'm kind of using Jack's niches talk that he did. And most of you guys have probably seen that, but I'm going to run through here. We got a picture from Bucks, this Squamanita personii here. Do you have anything you can tell us about this, Buck? Because
found it. Came to the fall show, if you had a basket full of
And-
beautiful Russian and micropus in the middle of it. It's that thing, and I said, that's too bad to put it out on the table.
Understandable.
Whoever actually was there did it. You're absolutely right, and it's a sick question, this one, look at later, they don't say anything like it. And it's parasitizes, Cystoderma amyothyma, and it grows on it, and it has a rotten smell, I totally caught it wrong, and that's the smell of rotten.
Toilet gone wrong.
There's some nice stuff.
I've heard like burning tires, but toilet gone wrong sounds good.
And, it was just, it was only the fourth or fifth ever found, 4 or 5 are from Cornwall, England.
Right.
And there's one from Japan, since this was the first one from North America, I think. I think it's been found a couple of times since then, but it's very rare, so.
It's very rare.
It's named after the British mycologist Pearson.
What does it grow on?
It grows on Cystoderma amyothyma. Yeah, it decomposes the mushrooms. Yeah.
Yeah, powder cap or something like that is the
So
name.
anyway, it's quite rare.
I've got more photos and maybe potentially a updated taxonomy for that. I'm not sure.
Keep hitting me up with bits and pieces to publish their own monograph, so it's slowly here.
You've been parting it out for decades now. So yeah, we've got all of these things grow on top of fungi. This Clitosobe sclerotoidea, all of this mass down here is compressed helvellas. If you've seen helvellas, the gray, really bizarre looking stipes, saddled tops. Dendrocolibia grows on blackening rusula is generally what people say it grows on. I haven't seen it necessarily just with that, but. And then this Melanospora lagenaria, however you want to say that, is extremely tiny, growing with Trichoderma pulvinatum, which you've seen probably also on these belted polypores. They're parasitites. This Antrodiella is a ICUN endangered species. You'll find it very occasionally, but really striking yellow. I'll keep moving here. Bryophyllus fungi, there's fungi that just grow on bryophytes, which are mosses or other small non-vascular plants. This Bryoblossom is really on the top of my list of things that I want to see. And Justin Pollin's taking a great photo here. These 3 basidiomycetes you could regularly see in the Northwest. And really a lot of these photos are Northwest centric or West Coast centric. There's really only 1 or 2 species that are from elsewhere in the world in this whole presentation. Bryocyphus, tiny little ascomycete, as is this Pisiza. Both I took as far as I could in microscopy, couldn't figure out what they were, and really only came to a conclusion after barcoding, which was, a way that getting into barcoding has expanded my tool set to get to identifications. Resinicolous fungi. I tried to find some of these today. Usually on any conifer that has a resin scar, you can find at least Xiphia resinae, which is extremely common despite being really rarely observed on iNaturalist. All of these are extremely small. You have to get really close. If your eyesight isn't sharp, you'll probably have to use a loupe. But they're all out there. They're really pretty common to see at least 1 or 2 of them on a hike. I see this Eustilbum and the Xiphia most common. Soricide probably third. Barely a hike goes by where I don't see one of these if I just look for them, essentially. Rust fungi. What an incredible family here. All of these rusts have really, I'm not going to talk you through this, but just notice that there's 2 host plants for all of these. So we've got these bizarre looking fruit bodies that release spores. They're trying to find a secondary host. And there's a lot of different morphological series of events that help them proliferate across the landscape, but pretty incredible. when I go out because they're tiny. And there's so many things that look like them, tiny pieces of lichen, bird poop. So many things that you look at, you're like, is that a cordyceps? And it's not, it's just some trash that somebody threw out or something like that. But here's some of my highlights from the last year. This cordyceps species PNW01, that indicates that it's a we call it a bin number. It's not described. And we just kind of assigned it this name. Danny Miller gave this name. So it's not close to any specific species, so we just keep it as species. PNW01 means that for the genus Cordyceps, this is the first undescribed species in our list. Essentially, you'll see this a lot. If you were watching the photos at the beginning of the presentation, there's a ton of those in there. This is my pet Cordyceps. He lives in my kitchen in a little Tupperware. And he's looking great right now. We're getting really large fruit body tips to all of these. But all of these entomopathogenic attack different arthropods and insects. Some of those are more clear. Underneath this Gibellula, there's a tiny spider. They seem enigmatic and rare. Whenever you kind of tune into them, you'll start to see them all over the place all year round, through the winter, through the summer. Most of the species are found tropically or subtropically, but our species, you can really find them. In the dead of winter, I found the most this year. Hypogeus and gastroid. This is probably the biggest group that I've treated with this. And they're all from different families. These aren't all related to each other in any sort of way. This is from right here on the Western campus that Jack found. I think a club member alerted him to that. And we're covering everything from puffballs. This is a Cortinarius that's going hypogeus. What's happening here is essentially an agaric, or a regular mushroom, is turning itself into a truffleized version, an underground version. It can't eject spores anymore, so it's relying on animals or something else to disperse the spore mass. And there's all kinds of different ways you can do that, either by smelling really good to animals and luring them in, or all of these glebal stinkhorn families types are kind of fungi that went to a hypogeous form and now are trying to form fruit bodies again and disperse their spores back above ground. And they are truly some of the most bizarre things that you can see really anywhere in the world. The ends of these kind of phallic fruit bodies will have a really strong scent. And by the time that these are done fruiting, really flies and things that would be attracted to rotting material or dead animals will have come and taken all that glebal material away, dispersing it. All of these are West Coast or Northwest species. This psilocybe is from New Zealand, but I think you could probably find it in the Northwest soon. Whenever you run ITS on these, which is a barcoding sequence, they come back as psilocybe cyanescens, the wavy cap. They have exactly the same psilocybe cyanescens. So without using other regions, and I'm not sure if other regions have been used, we can't tell them apart genetically from cyanescens. They're exactly the same. Coprophilus fungi, my girlfriend's least favorite. Because whenever you're crouched over dung or fecal matter on a trail taking photos of it, people really tend to not appreciate what you're doing or understand. Anyways, they're very interesting family, in my opinion. This pilobolus, this little spore cap on the end, it uses sunlight and this, I can't remember the name of this big spherical dome, but it orients itself to the sun using this ocular kind of sphere. And then whenever the turgor pressure is right inside the fruit body, shoots those black caps off at an unbelievable speed. I think they're technically the fastest living organism on the planet. They go from like 0 to whatever terminal velocity in like 0.2 seconds or something like that. It's absurd. Laziobolus, you'll see on a lot of herbivore dung. This Anthroderma is something I found last year on the east side in a snowmelt zone. So it might be a snowmelt coprophilous fungi. I'm not sure. It took a long time to find out what this is. A friend of mine who's Everyman Bio on Instagram, I can't remember what his name is off the top of my head, because I never met him. Anyways, Josh McGinnis. Yeah. Big thanks to Josh McGinnis for this. He put it into culture. He sequenced, barcoded a bunch of different fungi that came up in culture. And then eventually we landed on Anthroderma silverae. Here it is in culture. You can kind of see that it has some of the same characteristics. You know, all of these other fungi, we don't need to go into individually, but I found this Amerisolopsis on the foray that we did together last summer. So I thought I'd include that. It's kind of, yeah, kind of a Northwest mushroomer's special. And then this is another of my favorite, and this is the last of the ones that we're gonna use, but and Mycota is another name for snowmelt fungi. And this is a good opportunity to just talk about seasonality and like what that means necessarily, because you can find these running up the snow line all summer long. A lot of these I found in July or August. You hear snowmelt fungi, you might think is just a spring phenomenon. But you know, spring starts earlier at sea level, and then it progressively goes up in elevation throughout the summer as those conditions, you know, reach different elevations. So you can really find a lot of these all summer. Pseudoampelina angelestiana, I think, is one of the most beautiful mushrooms I've ever seen. And it's readily found in snowbanks. And all of these are also found well after the snow is gone in many cases, just maturing afterwards. And what's happening here, none of these are really super related to each other. It's just that the cold breaks and there's a big flux of moisture into the ground. So all of these are reacting to a new available resource and really taking off. Chlorocypha flavida is probably the most uncommon of these. I've only seen it once or twice. Hygrophorus goetzii is also really gorgeous, and all you need for that is to be around 3,500 feet or above where there's snow and there's mountain hemlock, and that's really all you need to know to find it. But another interesting thing about snowmelt fungi is like there's snow melting all over the planet, and not all areas have fungi that are specifically following the snowmelt edge. It seems to be kind of a West Coast phenomenon as far as I know. So something to be aware of since we all live in this area. It's something that we can participate in all through the summer, essentially. Okay, so that was kind of an eye candy section. We're going to move on to just some more cut and dried subjects here. Collection. While you're out, supposedly you have permission to collect wherever you're going. If not, use your own discretion. But I use these tackle boxes, and the way I load them, which this arrow is showing, is just wherever the different gates are that separate things. So like this is the first thing I collected in the day. This is the last thing I collected. If there's multiple days worth of stuff in a tackle box, I'll put a little note. If there's things that look really similar, I'll make a small field note and drop that into the box with them. And that just helps me keep things clear until I get them home, until I get them into a dehydrator. You're gonna want some sort of ruler or metric that you can put into your photo as a scale object. Allegedly, these gray rulers give you perfect white balance, but they really didn't do that in this photo at all. Another great thing to have is a UV light. I've got mine here. I don't really carry it with me in the field as much as it says fieldwork essentials, because most of the chemicals that are going to be UV active are still going to be there after something dries. When the mushroom dries, most of it's leaving as water, and so the other compounds are generally there. I don't know if that's true for everything. But you can do those at home generally and take those photos at home. The Ulanzi lights that you see here, I use these to light up a subject. In front of me, you can change the color temperature, you can change the brightness of them. And you can also use them for like different colored like rave lights back at your campsite afterwards too. And I use an Olympus TG6, which I have here. If anybody wants to look at it after the talk, you can come up and talk to me about it. It says TG7 here. We'll just ignore that. It's a completely waterproof, shockproof camera, essentially. I've dropped it down mountains. I've gone snorkeling with it without a case. I've used it out in the rain any number of times. It's really super durable. Takes pretty good pictures, but there is a kind of a small sensor. That's the trade-off for all of the other features that it has. It's really focused on macro photography. So pretty much all the photos that I had in the slideshow and what I'm using in here are taken with this Olympus, and for what it costs and what other things it provides you, it's a great field camera. Fundus, which is a organization out of California that has professional field workers, this is kind of their baseline camera. But if you get one, bring some batteries with you because they really suck up batteries. Also, things you can use in the field to keep your collections all separated are these project-based tags. These are Fundus tags, who I was just talking about. Mandy Hackney and Tabor are both really talented Fundus collectors. And this helps you keep your sequences organized all the way through barcoding and then into perpetuity afterwards. You see they've got little codes on here. You can scan that. That'll take you to the observation. That'll get you all kinds of places. A basic kind of chemical field test you can do is KOH, potassium hydroxide. And I just picked this one because the hot pink is really the coolest KOH reaction I know of. I think technically this would be a negative KOH reaction because there's a specific color that KOH is supposed to react. I can't remember what that is. I just describe whatever the color is. You can put it on the cap, you can put it on the gill structure. Sometimes different colors will happen. And I just notate those on my field observations. And so this is a good reason to bring a UV light into the field because you can get pictures of stuff like this just in tiny holes that are really incredible colors. I have some of this in the box actually. If you want to look at this, we can shine a UV light on it. But here's, Hyphiloma fasciculare. You've probably all seen growing out of stumps and logs around here. Under UV, it's super reactive. And you should bring a little field notebook with you to jot down notes. I tried to put a picture in here, but it was more boring than the text box, so I just left it. So more about the TG6. Here's some kind of technical stuff about that. But like I was showing you earlier, I use a low ISO setting and a dark aperture. And then I'm flooding the subject with light. Sometimes I'll turn these lights up really bright, especially if I'm zoomed in, and that replaces the flash for this kind of photography. And you can exert a lot of control just by backing up the lights, bringing them forward. If you want to look at this, I'm happy to send it to you, but this applies to more people, I think, because for years I just used my phone. And you can get really excellent photos with your phone, actually. There's no reason you have to have a specialized camera. What I would suggest, and I think this is true for Android and other kinds of phones, that you can hold down where you want the camera to focus. And if you hold that down, it will lock the settings of your brightness and your white balance. Generally for an iPhone, I turn that down. There's this sun icon. You can scroll down on it. You can see just between these two photos that in the greens back here, there's more variety of greens. It's a little more washed out here. There's just more depth of color. There's more depth of shadow, more contrast, and that just means more detail. So generally what I do is I'll hold my phone up to something. I'll hold that down, scroll it back, and then just pull my phone back slightly. And you'll notice that you can focus on anything that you want to, any fine detail. Clip-on macro lenses are great. That makes your range of focus even more narrow. And I've lost like 6 or 7. So really the moral of the story is to just buy the cheaper ones because there's like $100 ones. And I showed you the other two lights I use. This is the Ulanzi bar light. I guess I'm the brand ambassador. But this one is particularly good for cell phone because you only have one hand and you're getting a lot more directional light from it.
How's the battery life?
The battery life on these is actually really incredible. This one, not so good. This one, I'm using it at, 8 to 20%. I can use it for 5 or 6 days before I have to recharge it. I've gotten these soaked. I've dropped them in rivers. They work really incredibly, and they cost about, I think, $24 each. So they're not really cheap, but for what they've done for me and improving my photography, I recommend them. This was supposed to be a slide about like really high-end photography with nice cameras. I don't know as much about that, so I just turned it into people whose photography I've really appreciated over the years. This is Connor Dooley right here. Connor's probably taught me more about diversity of macrofungi than anyone else I know, actually. So I don't know how he found me on iNaturalist, but he seemed to kind of just adopt me and point me in the right direction. And that meant more to me than anything. Someone that just is kind of like patient with you, not necessarily knowing what you're taking photos of, but like helping you appreciate biodiversity. I've never met Ellison Pollock, so no gold star, but I'm sure as soon as I meet her, she'll have one. I've never met Stu or his beautiful dog. Stu might get a gold star later. Anyways, here's Jack Johnson, Lauren Ray, Connor again, Aiden Hirsch, who's also a Bellinghamite. And this is my friend Warren Cardamona, who I've hung out with a few times, takes great photos. Anyways, the moral of this story is find people to go mycologize with and build relationships, be outside together. It's fun to learn from each other and appreciate things together. So this here is kind of just to illustrate, like this is like what you want from a layout of Basidiocarps, like, regular mushroom morphologies, a group of mushrooms all together in their habitat with some still intact. But I think it's really important to include a lot of different environment photos, like just slight zooms back will often capture details that your up close photos don't have, or a few other things in the environment, like a little cone or something that you can come back later when you're making field notes afterwards and help you describe what the tree species are nearby, or like what the mid-story is. Like for instance, this is a dead Scotch broom and a baby white pine. And you can't tell that from this photo whatsoever. You might be able to tell that's Scotch broom, but also you'll end up with a lot of really nice landscape photos over the years if you just take a environment photo every time you find something that you think is interesting. So we're back from our field trip now, and we have to do some of the boring stuff, like vanilla our fungi into a dehydrator. What I would suggest is tagging things that might dry in a way that would make them hard to discern from something else. I just use a little piece of paper and some painter's tape. Generally speaking, that's never failed me. If something is really small and you're into Petri dishes at your house, use those. But what I use is just these to-go container, like sauce things, essentially. And if something's really small and you think it's going to dry imperceptibly, and you have PCR tubes, which is what we use to run barcoding, just put a little piece in there, because if it sucks back into a log of the tiny little ascomycete, you won't be able to sample it probably. Here's what my bags look like. I have close to 1,000 of these in my house. But what's important is you get the iNaturalist number. Locality is great, date is great, because somebody's going to ask you for a sample that you have at some point. And if you can't find it, then what was the point of collecting it in the first place? I don't know how to jump out of this. I was going to show you guys my Fungarium document, but essentially here it is. You don't really need to see a spreadsheet anyways. Some people have gardens. I have these spreadsheets that I live inside of and populate and just organize constantly. We'll revisit another one later. So you've collected, you've vouchered, you've dried things, you've got your sample. Maybe you did identification before, but this is where we're at now. So this is something I found on Monday, just walking my dog in the morning. You know, here we are around the stump. No particular gill color. But we've got some details here. We've got fine fibrils on the cap. We did a spore print. It was pink. I went and did microscopy, and I don't have a down lens camera, so I just used Michael Quo mushroom expert photos here. But we fit all of these categories. This is kind of how we would go about just a general identification. Not everything needs to be barcoded to find out what it is. There's all kinds of different physical attributes that you can use to narrow down what you're looking at. This is a selection from Mushrooms of the Redwood Coast that has a ton of information about what you should be looking for. And essentially what it says in here is that, like, I found this in an urbane setting and kind of a wood chips scenario. But what I want to draw your attention to is that it's important to make a detailed description of your collections. It gives your observation really the most impact possible as far as having a broad range of available information. If you get this barcoded afterwards, we've got spore measurements, we've got all kinds of things that will help discern between other species. So I'll just read the description real quickly. You don't have to use really intense mycological language necessarily. You can kind of use plain speaking for these descriptions. It makes it easier to generate and easier to understand. If you're making descriptions of microscopy, though, you're probably going to get into a lot more detailed language. I've got some resources for that in a few slides.
Question,
Yeah,
you've mentioned barcoding several times.
yes.
Who assigns a barcode to a particular mushroom?
So a barcode just means that we have genetically sequenced a certain region. And so it's a barcode. It's not sequencing would be, if you sequence the whole genome, you would have the entire sequence. So barcoding, mostly for fungi, we use ITS or we use LSU. These are different regions inside of the genome that we use that tend to have indicative sequences that we can use comparatively to figure out what's different about things.
Okay, so suppose I find a mushroom.
Mm-hmm.
Where do I go from there to find the barcode?
You would have to find someone who's doing sequencing work that you can get that sample into their barcode. So you would want to voucher things like I was talking about a sequence or two or a screen or two ago, get it in a bag, find someone who's available for it. I'm going to talk about this quite a bit more. But yes, a barcode is just a tiny region of sequence. I call it barcoding because it's really just a specific region. So let me read this description just real quick. It's we found scattered sporocarps, which just means individual mushrooms, near a decaying tree stump growing from soil and wood chips, seemingly saprobic, which means that it's just consuming and digesting like dead material, more or less. White to pale buff cap, approximately 10 centimeters across with a slightly incurved margin. It's incurving right here. Variegated light brown center, which means as we're approaching the middle, the color is starting to change, get blotchier. And then darkening towards the center of the cap, where dark brown and black squamules are apparent, which just means these little hairs right here. The gills are free, which means that they're not attached to the stem right here on the inside. And the gill surface is white to buff when fresh, but it gave us pink spore print. So as it matures, the gills become pink. The biggest sporocarp was around 8 centimeters tall, mild fungal taste, which is pretty normal. Things taste kind of like mushrooms from the store, but you should spit them out because they're not those. And there's a negative KOH reaction. I didn't show that on the cap, but there's no KOH reaction. Microscopy in this club, you guys have a lot of folks who are talented in microscopy. It's not my strong suit. I'm trying to get better at it. But in lieu of my lack of expertise, there's a really good instructional PDF specifically on fungal microscopy. If you want that link, let me know after the talk and I'll just email it to you immediately. There's a few things where you can just look at the spores, like this Asterophora, which means star-shaped spores or something like that, I think. And you'll know what it is essentially just from that. You still need to measure them to find out what species it is inside of a genus. But most things aren't as cut and dried as that. You'll need to take spore measurements, look at a lot of other features on spore surfaces or gill surfaces all over the mushroom to have an exhaustive microscopy going on. So there's two families inside of fungi that are kind of the main divisions. Basidiomyces, which is more your classic mushroom looking, and then Ascomyces, which is like your morels, cup fungi, helvellas, and things like that. And basidiospores means that they're attached to these basidia right here. These are individual spores, and they're attached to the basidia. They'll eject into space from there. Ascomycetes have these ascospores inside of asci, which are these longer tube-like ornamentations. So those are just two basic microscopy divisions. If you didn't already know, you could get through really quickly just by putting something on a slide. And if you have a downscope camera, you can automatically measure spores and other different ornamentations, which saves you a ton of time. Otherwise, you have to use a reticule, which is just kind of measurements that go across one of the eyes of your microscope. And then depending on which magnification you're using, you have to do an equation to find out what the actual measurement is. This is so much easier. This program is called Pixometer. It's a French company that a lot of folks use. I think there's other ones now. Okay, iNaturalist. It's kind of like my Bible in a lot of ways. I spend more time learning about fungi on iNaturalist than just about any other single resource by a long shot. And there's a lot of things that you can do with it. I can't figure out how to click out of this presentation, so I'll probably skip some of that and just describe some of the more powerful things you can do with the platform. There is an iPhone version, which is great. You can observe things in the field, put them straight up. Generally speaking, I use the computer PC platform. It's orders of magnitude more powerful. You have access to projects. You can go to individual observers pages, look through all of their observations, make comments, click through, add different, a lot of different categories to them. So, yeah.
On the PC, are you referring to a browser or is there an actual app that you can download this?
It's a browser, just the iNaturalist.com, and you can log in with the same iNaturalist you use. But the suite of tools that are available to you are tenfold greater probably on the web platform. There's so many features on the phone app that you just can't access at all, including projects, which are a great way to learn about specific fields of interest. A lot of things from the slides I was showing you earlier about, specific environmental areas, there are projects for. There's snowmelt fungi, there's peatland sphagnum fungi, which are going to be the bog and fen fungi, resinicolous fungi. And a lot of these you'll click into the project and it'll show you what the parameters of the project are. And that will be an exhaustive list of all the species that you could expect to find somewhere. So if you're going to a bog this weekend, you've never been to one before, you kind of want to know what you might encounter, you can go to Connor's peatland and sphagnum fungi and see a pretty exhaustive list. I mean, other fungi can happen there, but things that are, dependent on that environment are all going to be pre-listed here. This is an example of a PSMS project that I've been working on with Shannon Adams and Kendra Dudinsky and a lot of other folks show that's here. And every time the Myco Blitz comes around, which is run by Mycoto, which is Stephen Russell's barcoding workflow, essentially. He'll put on these events every year, which like for a week, you can find and voucher any fungi that you want, send in 10 fungi, he'll sequence them for you for free. So we made a project out of this, we've, sent more samples than that to him. But we've gotten great data from it. It's a great resource. I included that because it's something that I've been working on. And then just some basic guidelines for iNaturalist. You're gonna want to take a photo of multiple angles of each mushroom. You're gonna have all the different surfaces that are important. Your gill surface, if it's an amanita or something, you're going to show the annulus. You're gonna want to show the bulb at the base. There's all kinds of different factors that you'd use for identification. And then there's observation fields, which we're gonna touch on right after this. And I don't do this nearly as often as I should, but you can make a really high quality iNaturalist observation by just adding a few of these really detailed categories. You can also use those categories and left click on them. I'll show you them in a second. And then you can search every observation across iNaturalist that has that category, either specifically with the same result or just broadly using the same category. If you're making identifications on iNaturalist, for yourself and for other people, I would just suggest that you be a little bit conservative, leaving things at a family or a genus level, which are, a step or 2 steps below, species level identification is really ideal unless you can demonstrate that it is specifically the species. If you have data to back that up, if you have specific microscopy or something else that confirms, within a relative shadow of a doubt that that's what you have, it's preferable just to kind of leave it at a lower level. It says here, unsubstantiated claims dilute the scientific value of the platform. But if you looked at iNaturalist, there's all kinds of crazy blurred images that are not anything so you can't really dilute it too much. But so here's a great example of observation fields. Dean Lyons is a field collector for Fundus, which is the same California organization. They have a bunch of people that work in the field who are paid to collect fungi and send them in to Hartsinger and other people to both sequence and then interpret the sequences afterwards. They're a great group of people, all doing really high quality work. I know many of them. I have not met Dean, but I'm looking forward to it. Dean gets a gold star because look at these observation fields. There's 16 of them. They're highly detailed. You can see what some of them are and how they would be useful. The accession number is using this tag here. So we're using our tag to both organize it in iNaturalist and through the barcoding workflow. These are the whoa. DNA barcode ITS. That's the results that we got back from the barcode. So you're asking about a barcode earlier. This is the result. It's in the ITS region, which is just a small, like 600 to 800 BP region. We got microhabitat, fruit body abundance, general description of the location, which is like the trail that you would be on. KOH reaction, notes, provisional species name, which we were talking about earlier. This is closest to Xeromphalina cornui, and it's in the genus Xeromphalina. It's the 3rd unknown genus Xeromphalina that's been observed. So it's cornui PNW03. Reads and consensus is some jargon from nanopore barcoding. There's nanopore, it's a bunch of small pores, which is what the name suggests. Reads and consensus means that's the number of pores that attach to the sequence and read and called back ITS data. And then those are compiled. That's pretty accurate? The sequencing is nanopore. ONT means Oxford Nanopore Technologies. It's just a specific company. Substrate trace files are the raw files that you get back from the lab. And all of this together makes this a really high value observation. If it was just Xeromphalina cornui and this ITS data, we wouldn't know all of this without going back and specifically asking someone about it. And then before I move forward, I just have this little quote from Dean that's on his iNat page that, yeah, I agree with, but you can take or leave it. Okay, next we have Sadie Hickey. getting a gold star for description of specimens. Can you read these? No, not really. I should have just used a few of them.
Can't even see the cursor.
Can't even see anything, yeah. So Sadie uses really just common parlance descriptions. This Intelomataceae is growing in rich exposed soil in Darlingtonia californica meadow. Pileus blue-gray, frosty textured with a distinct umbo turning lighter and more brown in age. Lamellae off-white, narrowly attached. Stipe whitish, fibrillous, twisted, growing crowded. That's exactly what's happening here. So you don't have to go into using really bizarre mycology words, even though they're really fun to use when used properly. But a description of specimen is another baseline for a really high quality observation. The better your photography, obviously, the more visual alert it's going to have. Stu Pickle, I'm not sure because I've never met him, really masters this. I mean, these images are well above standard and just gorgeous. This is some Intelomataceae. This is in Pisiza, like the cups. This maybe it's in Satharella. I'm not sure if it's the aquatic Satharella. This is a UV reaction. This is the same Amerisolopsis that I showed you earlier, but my photo pales in comparison. Arachnopisiza, Melanophyllum. Just really gorgeous photography. That all helps. So now we're going to move on to just some resources that you can use in identification or just to learn more that are outside of iNaturalist. Fungi of Temperate Europe, volumes 1 and 2. This is probably the best book I've ever seen for macrofungi. It's I'm going to show you in a second. There's so much information just kind of coalesced into one space. And if you check this out, I can also send this to you, this link. There's 92 pages of these wheels, which just have an insane amount of visual information. This is, the broad category of agarics. We've got tons of families here, all kinds of different species, in a concentrated visual space. We're showing you microscopic, features, and they're kind of organized phylogenetically. They're kind of near groups that they're related to. We've got hygrossivoids, beautiful group. We've got a lot of information here. These little, gill caveat photos here. There's just so much information, and it's just a stunning book. It's really expensive, is the caveat for that, but they're about to make a new edition. So wait a few months if you want to buy this. And the first edition should come down in price quite a bit. Yes, the wheels are incredible. They're free. They're there. They're not for the Northwest, but they're telling you so much information that is translatable just for, visual differentiation. Colibioids, I've put this one in for Buck back there. This is a family that's near and dear to me. I spent a lot of time with entelomatoids. There's just a huge wealth of information inside of the wheels for just about every family is included in the wheels. They don't go into the species by species breakdown. You have to buy the book for that. These, however, are regionally specific. I'm sure a ton of you know about mushrooms of the Redwood Coast. Mushrooms of Cascadia is coming out this year. Does anyone know?
September 1st.
September 1st, mark your calendars. This book has been out for quite a while. I assume it was made physically, but I've never seen a physical copy of it. But it is also incredible. They're both made largely by Noah Siegel and Christian Schwartz. Elsa Valinga is heavily involved in this, as is Michael Castellano, who's an expert in hypogeous truffleized fungi. And I don't know who Diana Queta is, but they've done great work in that book as well. You can find that it's out there. Here's some other books that I use. That doesn't mean that you need to use them. This book by David Largent, who is a regional entelomatoid expert. This book is great if you're interested in entelomas, but also if you want to learn microscopy terminology just through going through somebody else's study cases. There's a lot of great things here that this book is also by David Largent and David Johnson, and covers a lot of that material. I found them both useful. Polypores of British Columbia is just a polypore guide that was made by the government of British Columbia. There's a link to it if you want it. I'll happily send it to you. Macrolichens is great. It's the Bruce McCune book. There's several volumes that get into. Okay, so this one really just shows you fruticose lichens, which are like kind of leafy or more.
That one just shows you natural lichens.
Yeah, the larger foliose lichens. Yeah. So things that are like on rocks or like they are not going to be covered here. So no gold star, but just because there's a limited range of genera. This glossary of mycology has every word I've ever heard used inside of mycology, and it's just a great reference. There's so many things that I learned about just pronunciation and all kinds of other features from that. And then Ascomycete fungi in North America by Michael Bugh is great. The Bassett's also make great guides for things in well, all over the country. But all really great books. Web resources, they're free. We should use them. There's so much to learn. Northwest Mushroomers Association has a group that a lot of you are active in. It's a great place to take your most recent photo, get second opinions on it. You have a question about, growing mushrooms in your garden or something like that. I've seen as a recent topic. You can go and talk with your peers. And then, once you've met each other online, you see each other at a meeting, go say hi and build a little bit of community there. This I gave a gold star because Danny Miller's website, the Pictorial Key for Mushrooms of the Pacific Northwest, has an insane amount of genera listed on it, all with great photos, great descriptions, and all of the other bin number fungi that we've been talking about there, undescribed, are all listed really on Danny's DNA Discoveries. He establishes a lot of the P&W numbers that you're going to see on iNaturalist, and almost all of that data is now here on his website. Ascomycetes aren't covered as heavily as agarics, but he's just done a big update. There's a lot of stuff on there. I haven't even actually looked at what's on there now. There's Mushroom Observer, which is like kind of another social platform similar to iNaturalist. I don't use it as heavily, but a lot of folks love it. Mushroom Expert, I used the microscopy photos from Michael Koh's website earlier. And that's a great resource for just going to, you found something, you think you know what it is, it's going to list the microscopic features. There's going to be a good description of it. And then does anyone use Matchmaker?
Yeah.
Or MycoMatch? Another great resource. It's really, really great. I mean, like you can narrow down what you have material-wise with just a few clicks on your screen. And there's a ton of taxa on there, which is just, taxa just means like a species name, effectively. And then there's keys, dichotomous keys, where, before we had these really nice books with a ton of photographs and detailed knowledge, you would just key something out. And for the Northwest Key Council has an incredible amount of key resources. And then I am constantly watching presentations, mostly on YouTube. There's some great ones here. I had to reference Jack's Cryptic Niches talk because I kind of used some of that visually earlier. And then Lauren's talk on hygroscopy is incredible. Noah's talks, then there's so many available online. Great, like kind of digested information. So back to some cut and dried stuff. Phylogeny and taxonomy jargon. This isn't really my strong suit, delineating some of this stuff, so I kind of just tapped this out. Taxonomy is a hierarchical classification of species, like you see that here, where hierarchies moving back in time. And then every species level designation gets a binomial name composed of a noun and adjective, which a lot of people call scientific names. A good example is Agaricus bisporus, which is the grocery store mushroom. And its name, agaricus, is Latin for mushroom with gills, and bisporous means that it has 2 spores per basidia, which we looked at just a moment ago. So a really descriptive name. When you start naming things after people or what other binomial things get included, and it doesn't tell you about the mushroom, I tend to like names considerably less. Phylogeny is an evolutionary tree right here on the right. That's a graphic representation illustrating evolutionary history between a set of species in context of time. So from left to right, this would be the oldest species evolutionarily. It branched here into these subgroups. And then a clade is any branch or sub-branch group. It's like A and B are a clade here. A, B, C, and D are a clade here. And notice that there's a lot of these nodes here that aren't named. Those would maybe be subgenuses or something like that if you're talking about a species or a family that's been divided. We use clades and phylogeny a lot in barcoding to organize data visually, tell you about who and what are related to each other. So now we're going to move on into the sequencing technologies portion. And Sharon did a presentation on this, what, a year and a half ago? So I'll give it kind of a light treatment. This is what you would have at your house if you wanted to have a PCR home setup, plus a few more things. These are my micropipettes. We'll be talking about how to use those. These are the tips. You just jam a micropipette in this box. There's a tip with a filter on it. You can suck up liquids with it and move things across these tubes. Once you've gone through your PCR protocol, you end up with a PCR product, which is represented here. And you can send that either to a lab for Sanger sequencing, or you can send it off for nanopore sequencing. You would just have to use slightly different reagents, different volumes. And so if you have PCR product, you want to send it off for Sanger sequencing. A lot of folks use GeneWiz. I use MC Lab because it's in California. It's a little bit cheaper. It's really cost effective, actually. And then nanopore sequencing is maybe a bit more detailed. We were talking about reads and consensus earlier. It gives you a ton of reads and then kind of coalesces them into an average file. You can go look at and see if there's, single BP or different, and then tell, what's going on between species that are closely aligned. You can send in a PCR product or just tissue to Mycology, who we talked about earlier, Fundus, and then Ohio Mushroom Lab. You can, without there being a mycoblitz or anything, you can just send in tissue, and there's no saying when you're going to get results back. But Kyle Kanan is just open the doors wide. You can send in whatever you want. Eventually, you'll get data back. That's kind of the thing with nanopore sequencing. You have a high volume of samples coming in. So you're sending samples off. You're not really sure the timeline that you're going to get data back. You will likely get really high quality data back, though, when you get it back. So there's also these next generation technologies, which I don't completely understand, but they're really incredible. What's going to happen with these? Once these start proliferating, the prices of all the other sequencing technologies are going to plummet. Right now with Illumina, you can do a whole genome for fungi for about $1,000. And what it's doing, you can see here, this is an Illumina sequence. It's taking a bunch of short reads and then combining them all together to give you a really long file with a ton of data in it. And then these packed biosystems, nobody has these at their house. They're really just institutional, but they will run multiple regions, like 5, 6 regions all at once, and then give you all that data back. So instead of just getting ITS data, you're getting really, really diagnostic information, the highest quality information that you can currently, essentially. And it also does it all automated. You don't have to pipette things. It's pretty incredible. And the way a lot of these technologies work, and this is kind of an image of nanopores, they're really just physically extremely tiny pores with these oligos sticking out of them. and you'll do what's called library preparation. You'll take your sequences, append something that will attach onto these oligos that will pull it through the sequencing membranes. And then it calls back the data in a similar way to what Sanger does. These Sanger sequences, they use, they'll put a fluorescent dye on them and then take a series of pictures, essentially a movie, and then call back what it saw on them, A, T, Gs, or Cs. And that's more or less what's happening with these nanopore technologies. So here we go, protocols and reagents. To run a PCR, you're gonna need a protocol that tells you just like what to do and what steps. I use the Dicaria protocol, which is made by Hart Singer, who's a sequencing coordinator for Fundus. He also has Dicaria LLC sells all of these different reagents for really, really affordable prices. And before there was a fungal specific reagent depot, essentially, which Hart has created, it was really hard to get your hands on some of these primers without having a business license or an academic affiliation with the school, because who wants to send chemical reagents to a stranger off the internet? But Hart has been super helpful for me in learning how to do this home barcoding stuff. And so I just wanted to say thank you for him. Extraction is step one. You're going to sanitize your workspace, which means just spraying it down with a bleach solution and wiping it down, get everything ready. And then you're going to sample. You can sample 8 things at a time with the equipment that I have. So I just write them down physically, and then I'll move that onto a spreadsheet later. But you take tiny samples like this. This looks like a really small sample. This is actually kind of a big piece of sample material. Take a tiny little piece of each and drop them into these tubes that you see here that we'll put the extraction liquid into. Then we're going to use a pipette. We're going to add the DNA extraction solution and then use a pipette kind of as like a mortar and pestle, crush up the material so that the extraction agent can start to lyse like the process of lysing genetic data out of the cell material. And then this is a thermocycler. Mine only has 8 wells. If you decide to get into home sequencing, MiniPCR has an 18 or a 16 well, and just buy the 16 well. It's so frustrating to only have 8 wells. I don't know why I did that. It's like $100 more or something. After you've gotten that, you need to dilute your samples because the extraction agent will just keep pulling DNA apart until you can't sequence it anymore. It'll be so fragmentary, it wouldn't be worth it. So you're going to dilute it after you run it through the thermocycler, and then put it in your centrifuge, spin it down. I use the 1:5 dilution for most things. There's a lot of families that are hard to get DNA from, or there will be other PCR inhibitors, like I detail here, things that are really gelatinous. Dirt is a PCR inhibitor. So you really want to have clean samples going into tubes as much as possible. The next step would be preparing a reaction. For PCR, you got another set of tubes. You mix some of those reagents we saw earlier. Queen Green Tech, TAQ, water, and then forward and reverse primers, which are gonna adhere to the front of the sample, and the back sample give you a read from the front to back, and then a read from the back to front. You can use the software to align those to make a contiguous sample. And then you've got a more robust sequence data coming out. Once you've got that spread out across the tubes, you're going to take a little bit of your template DNA, which is the extracted and diluted solution, and just a tiny bit of it. Add it to this, and then run a PCR based on the program that's detailed in the protocol, and that takes about 2 hours. You can see here we're almost at the end of our run. Well, you can't really because it's so small. We've been running for about 1 hour and 50 minutes, and we have 8,589,000,000 copies of the DNA that I started with. And I won't go into how that all works exactly because it's just really time consuming, but the DNA molecule is going to get unzipped at certain temperatures. And then the primers that you have are going to have all the little pieces that would attach ACs, Ts, and Gs are all you're going to get back. Each one of those has a complementary letter that's like the only generalized possible thing that can attach to it. So all those little pieces are going to come fill that back up. And then as that happens 30 times, you're getting, you know, a huge bell curve of copies being made to where at the end, you know, you're well over 8 billion like we see here. And then the next step is gel electrophoresis. So you're going to take a little bit of your PCR product and you're going to make a gel, which is right here, cover it with a solution of TBE buffer. And then you put a little tiny bit of your product into each one of these wells. And then whenever you electrify this, the current is going to pull the material through the gel. And since we've added a nucleic acid stain into the gel, you can actually visualize the DNA moving through. And that's how you can tell if you had a successful run or not, essentially. This is one that I did just this week. These two middle lanes are not super strong lines, but I'm probably going to send it off anyways, and I'll probably get good results. But this lane right here, number 4, failed. I sent it in and I didn't get any result from it. I got a really jumbled file. But this is just kind of a visualization of what these lanes are. This is 8 fungi that I ran all the way through the barcoding process. And then this is lane 1, this is lane 2, this is lane 3. You get it. And then you have to send your samples off to the lab, which is really kind of a painful process because you still have to manually type all this stuff in. The best way to do it for me has been to just put the genus name and the numbers from my station. The files back, that's what the file name is. And generally I can move from there. I have another beautiful spreadsheet that I've spent endless hours populating that I track all this in. It's really important to just manage your data as it comes back in the easily accessible. I can click into the files. I can't scroll over and show you, but the data is all copied and pasted in there. Links to the raw files are there. And then anyways, you have to keep your PCR products stored in the freezer until you're ready to send it off. And most freezers have an automatic thaw cycle where it'll heat up and kill all the frost, which will destroy your samples essentially. If they thaw and then refreeze, you're getting crystal shards or cutting up your DNA essentially. So I keep mine in this ice box with these freezer pads over them just to make sure that when that happens, everything's still cool. And then you have to label them according to the spreadsheet. You see these are different 8 sample sets here. And then you wrap them up, mail them off. And generally in 3 to 4 days, I get this sequence data back. This is a chromatogram, which is one of the 2 files that you get. There's tons of programs that you can use to read these. And a lot of times they'll just have these kind of like useless areas in the front that don't tell you very much. But these bars are giving you quality scores essentially about like how well each one of these bases, each individual base that you see is a GAGT, GACT. This is the order that the either Sanger sequencing or the nanopore called these sequences. And that's what we use for the barcodes. So you'll go through and sometimes there will be overlapping sequences. There's not really good ones here where you can't tell exactly which one it's meant to be. You use these codes to annotate those. And then you copy and paste that simply, come to BLAST and paste it into a window. BLAST cross-references your sequence data across tons of other uploaded data. And then you get these comparative results back, which you can then use to make your species level designation if you're so bold. You can't read this probably, but it says DNA analysis and sequence validation, a case study in glossing over so much detailed information that it's unimaginable. There's really just so much happening inside of these. Like we don't know who put these up on the internet. We don't know if they're accurate. Oftentimes they aren't. But there's all these different scores that you can use to see how close you are. This length that's how many bases that it was similar for. This is the percent of all those bases that were the same. And a higher score generally tells you that you're closer, but if that thing is mislabeled, then you're much closer to something that's wrong. So really when I got into barcoding, I thought I was going to have lots of clear-cut answers, but it doesn't work that way, sadly. There's a lot to unpack for all of these, and I'm really just starting to learn about that. I've only been barcoding at my house since September, and I'm not an expert by any stretch of the imagination. Anyways, since this is a case study, we're going through what happened with this specific species that I found in a park in British Columbia. It looks just like Marasmiellus candidus, really, but with these really bizarre gill patterns. But anyways, these are the results that I got back. And then clearly through bouncing those off of BLAST, it came back as one of the only sequences that matches this outside of China where it's described from. But it matches so clearly that a lot of experts that have reviewed it agree that that's what the species is. So after you've done that, you can just take that same data that you put into BLAST, copy that into the observation field. DNA barcode ITS is what we generally use. If you have data, you want to put it there so that, like I was saying earlier on iNaturalist, you can not left click there, but you can left click on observation fields, and then a drop down bar will come. You can say select all observations with this field and information or whatever. And if there were other species with this data, you could see them all next to each other. They just come up, they'd be next to each other, cross compatible. And then you would want to go to that observation, put in a comment, like a link to yours, say like, this appears to match your species. And right now, since they don't have species level names, that's the best way that we can track them. There's also an observation field in iNaturalist that is a provisional name, which is like what we're calling it. And that's where you would put like Omphalina favrii PNW 01 or something like that. You can also cross-reference those. They don't have to have DNA data, but you can see everything that we're calling this undescribed species all in one place. See. Yeah. I once I'm done with this, I will jump out and do a little bit of clicking through. I'm not really sure how to do that in this Zoom presentation. When I escape, it just brings this menu up. So I'm scared. I'm scared to go outside of it. Hide floating controls. Okay, the most absolute boring thing you can do with your data, and most important, is to put it on GenBank afterwards, which is what BLAST cross-references. There's a bunch of other databases too, but GenBank is really the main one. You can upload your sequence data. There's pretty stringent protocols for like how you should construct the name, so that you come up with something ideally like this one that has a voucher number in it that you could go look up on iNaturalist, and a bunch of other information that you can tell where it came from, who posted it, and just have access to it. And then eventually, as with just regular iNaturalist observations, somebody's going to come along and have a different opinion about what you thought something was. And it's important to just be polite and have a good attitude, because it's just a website, you know. It's data that you worked hard to get a hold of, but a lot of times they'll be more correct than you. And what we're trying to do is create community consensuses about what things are, so we can decide like what undescribed species, like what the boundaries for that are, what we should call it, what characteristics we use to reference that in the future. And then that will all help us describe these in the future. Or since we're getting data back so fast, way faster than we can describe species now, we may be able to just use these bin numbers for conservation, which is really the end goal of all of this. We're trying to get all of this knowledge so that we can have a baseline understanding of what our regional mycodiversity is. Once we know what all those things are, we can start describing what their ecology is. We can start using them if they're rare or if their habitat is vanishing, ideally to spearhead conservation efforts in ways that so far have really only been available for plants and animals, which is just not adequate. Fungi are a huge kingdom. They have vast and massive ecological import on what happens on our planet. So you've made it through all of the dry part, and we're back to pretty pictures now. This, as you can see, is my list of mushrooms that I want to see but haven't seen, in kind of a way that I can shine a light on photographers and other regional mycologists who do great work, see gorgeous things that I haven't seen yet, but that, you know, keep me excited about getting into the field. This Fabulosia grows on graminoids or grasses, mostly out on the coast, as far as I understand, out in Oregon. Dendrostabellus smaragdina grows on really decaying ABs, which is, you know, firs essentially. So small, really super gorgeous though. Cudonia grisea, there's a whole family. These are all really super gorgeous in my opinion. They're strange. Really looking forward to seeing this someday. Okay, this is a Hygrossibaceae, but or what's that family called? I really hope to see them. We're really just in Oregon and especially California. They're beautiful. What can we say? This picture was already, this is the only picture that's in the presentation quite twice, but it's so good, it deserves it. This is another species that just co-occurs with certain bryophyter mosses. Here's a picture. This, I learned about this from when Lauren and Sadie found this originally. And apparently this smells like burning tires or creosote, despite being just a gorgeous purple mushroom. And it happens sporadically up and down the coast. This is Claviceps purpureus is ergot, which is what LSD was derived from essentially. And it grows on ryegrasses all over the world, as far as I know. I haven't seen it yet, but I've been looking. Ophiocordyceps ravenelli. As Connor brought this into my awareness, but just a really gorgeous dune species inside of Cordycipitaceae that affects these little grubs, and really all you see sticking up out of the sand is just going to be these lighter tips, and you have to kind of dig down and excavate them. Also a coastal species. James Conway is a really excellent photographer, and I just got to meet him a few weeks ago. But he found this grub with just these little growths coming out of it and put it in a humidity chamber as a pet. And weeks later, these gorgeous, gorgeous growths come out of it. Here's Sutella. Mythicomyces corneopes. Someday, me and Jack thought we found this last summer, but we didn't. And here we're back to the same Squamanita that I posted earlier, but I think there's been an update to its name. I think it may be Disoderma from Fimbriata now, but you can see it's parasitizing these cystodermas, and it completely takes over their morphology and just grows inside of them and forces them out of the way. Here's a picture from Kit Skates Barnhart, who is the founder of the Key Council. Thank you, Jack. And then Connor's found it. Connor finds everything. Here's another, I'm not sure if these Cotylidia are the same species. I kind of imagine that they're not, but just these really beautiful fan shapes. This is the species that I really want to see with these kind of smoother sporocarps. This is kind of a parasitic fungi, and it's making what's called a pseudo flower, which looks kind of like a flower. It has similar compounds that make it smell like a flower and attract pollinators. Insane biology. Stereopsis humphrey grows just on the coast. I think it's associated only with Sitka spruce. I haven't seen it. It's in Victoria, apparently, on Vancouver Island. It's in Washington too. And then Brygioporus nobilissimus is just a huge polypore that's associated with old growth. And since most of our old growth has been logged now, it's mostly associated with old growth stumps, sadly. But you can still find it, it's still out there. And then we're ending kind of where we started earlier in wetland fungi. And this Sarcoleodia globosa is insane looking. Connor and Jordan both found these together last summer. This is the same sporocarp even. But beautiful. No clue what its phylogeny is like whatsoever. I'm not even gonna try to say this. We're not sure what it is even, but some just weird thing that Connor found and showed. And then Bogbodia uda. And these are, I'm not sure if either one of these are actually Bogbodia uda because there's no data for them, and they're so close to other things that you can find in a bog that are in the same family. But they must look really close to this. I haven't seen it. Connor has told me that, you know, that like you really are going to want to see data on that before you call it. So another thing to reinforce that you should just be conservative about what you call things on the internet. If you call it Bogbodia uda without
So
data.
what's the common name? Bog bodies?
Yeah, something like that. Yeah. I think it's so uncommon as to not have a common name, but it's really evocative. It's like it's close to the Hyphalomas. That's my talk. I've got some special thanks. My girlfriend Sarah Chu gets 3 gold stars. That's more than anyone else. And then my dog gets 1, but it's a really big one. I'd like to say thanks to Connor Dooley, Danny Miller, Lauren Ray, Jack Johnson, Sharon Squazzo, Hart Singer, Kendra Dudinsky, tons of other people. Joe, all of you all get gold stars in my book now that we've met each other. And thanks for having me. It's been a really great experience. My first talk, and thanks so much.
Will I be at Saturday's foray?
Will I be?
Yes.
Sadly, no. I'm at the end of my vacation time, and I just don't have it in me. No, I wish I could, but Washington Park is incredible, and I think anyone who goes will have a great time. There's interesting plant communities out there. There is a really cool Cooper Saceae out there that you can only see out there in Washington, and otherwise you'd have to be on Vancouver Island or in BC. There's cool stuff out there. You guys will have a great time. Somebody had a question?
Yeah, I do a lot of photography of mushrooms too, but I don't really publish it, put it out there tonight. But one of the issues I've got is color coordination or comparing colors from my photographs with
Right.
what I see online. And it's almost impossible.
Definitely.
So how do people deal with this if it's in the know or in the insiders?
Like how to make your colors align with what you're seeing in situ, or how to
People like that, it's just when you do a comparative.
right.
I've got special tools on my computer where I can capture any color from my screen. It'll tell me exact color representation.
Right, the color value number.
Yeah, color value. And, you know, it just doesn't seem to be working for me.
Yeah, I mean, color is another morphological characteristic, just like any of the other attributes that it would use, and they change over time. You know, as a fruit body matures, the color is going to change. So that's a hard part of comparing photos online, especially. Using like a grayscale ruler or something like that, that kind of gives you a nice white balance. There's also photo editing things, like a lot of people will turn the saturation on their photos up really high, so that like the purple of their Lacaria just like explodes out of the screen. But that's really misleading, and I hope people don't do that.
One of the problems with this is that every device that delivers an image to you delivers it differently. And one of the things you can do is calibrate your devices so that they're delivering its color that they, you know.
Yeah.
And
Carry a grayscale card with you.
well, this is different than that. It's actually, you buy a device that looks at the color on the screen and adjusts the screen so that it comes out correct. You're looking at color bars. It's been a while since I've color corrected my screen, but I did do that a while ago. Then when you get back, you've taken the photograph, your mind, if you have a pretty good color sense, can distinguish lots and lots of fine gradations of color. You want to adjust the color on your color calibrated screen to match what you saw in the field. And like Matt's saying, a lot of people will jack up, you know, the saturation.
Yeah, that's the
to bring
common.
out the garish colors that you see.
Yeah, you'll see all
which
kinds of
aren't true
other
at all.
background color textures pop out. It's really
Yeah.
degrades your image. But you can use the temperature of your light from like a yellow to white. That will greatly affect what the color coming through your camera is. That's kind of another white balance adjuster. I use a aperture setting that makes the base image darker and then flood with light. That tends to get a truer color image off of your sensor on your camera.
So do you take almost all your pictures with lights?
Yes. I use these lights. If I had a DSLR or, you know, one of those body cameras, I would use a flash with a diffuser. But the flash on this camera is not worth using essentially. And these lights, I can adjust how close they are. You know, I'll take a photo. It looks a bit dark. I'll bring the lights closer. And I'll take like 5 or 6 photos of the same thing with different aperture settings, different lights. And then I'll review them at home and pick the one that's the closest to how it looked in the field. There's also editing, you know, photo editing things you can do. You can raise the, yeah, almost every image you've seen in this is just raw from my camera because I have no patience for editing hundreds of images. But if you just, you know, boost the contrast a little bit, that'll give you a little bit more visual definition. It won't really adjust your color so much. You can turn down shadows and things like that. Also, to make things look more like they did in person, without just boosting the color information. But you should turn your saturation up probably just a little bit, like because your camera or white balance settings might not be giving you, or the sensor might not. For instance, the sensor on these TG cameras is not huge, and so sometimes I need to suggest that it gives a little bit more richness of color by adding just a tiny bit of saturation, but not much. Just a
I
little.
was amazed last Friday. I was taking pictures of the Northern Lights, which I hope everybody saw.
Oh yeah.
And with my iPhone, the iPhone, and it's probably true for all of our phones, has much more sensitivity to red than our eyes do. So these pictures were coming out just absolutely gorgeous.
Yeah,
But
that's
it wasn't
incredible.
what I was seeing.
Yeah, the night
Was-
the night settings made things so much better. And like honestly, iPhones give you a truer color more often than a more professional grade camera. If you're not really focusing in on settings and taking multiple images, kind of a spray and pray philosophy for me. I take a lot of photos, pick the one that looks the best, and then really don't do much post-production. Yeah.
Get a bother you about iNaturalist again.
Oh, please. Oh, you want to look at, well, let's look at iNaturalist.
Yeah,
How do I get out of here without stopping sharing?
because the sequencing stuff's rolling in, and because I'm really isolated on South Wood B, and I, don't hang out with people that do mushrooms ever, so.